Welcome back to our last video for Chapter 3 material, and we're going to be wrapping up Chapter 3 by looking at nucleic acids. So nucleic acids are our final type of biological macromolecule. We've gone over carbohydrates, lipids, proteins, and again, this one is our last one, nucleic acids. So why are nucleic acids important? This is because they make up the genetic material of all living organisms, and we've got two types. Deoxyribonucleic acid, or DNA, or ribonucleic acid, which is RNA. These are found in the nucleus of eukaryotic cells. We find them in mitochondria and chloroplasts of eukaryotic cells as well. Prokaryotic cells do not have a nucleus, so we will just see them in the cytoplasm, in the center or in the middle of the cell somewhere. All of the DNA in your cell is known as the genome of your cell. And DNA is very long and it's like a string-like substance. It reminds me of like the back of my TV or where our computers are, where there are a bunch of different cords, charger cords, computer cords, etc. Imagine you have all the string and you need to organize it. DNA in our cells are organized by wrapping the DNA around proteins called histone proteins. And this is true in, again, eukaryotic organisms like us. Prokaryotes don't have histone proteins, but they do have similar proteins that they use to organize their DNA. DNA, complexed with histone proteins together, is known as chromatin. And chromatin is further condensed to pack it tightly within the cell or the nucleus into structures we call chromosomes. Now, there are thousands of genes in our DNA, and genes are what contain the instructions for building proteins or other types of molecules like RNA in our cell. The DNA really controls all of the activities inside of the cell, and it does so by turning genes on and off. RNA, on the other hand, is mainly involved in the synthesis of proteins. And there are three types of RNA that are involved in the synthesis of proteins. The first one is messenger RNA, also known as mRNA. And this is really like an intermediate nucleic acid. I say intermediate because we start with DNA. DNA will go through a process called transcription in order to produce mRNA. And mRNA usually leaves the nucleus in eukaryotes in order to go through further changes to produce our eventual polypeptide and protein. Transfer RNA, or tRNA, is also involved in this process of translation, which serves as a bridge between nucleotides and the final amino acid chain. And then finally, ribosomal RNA is involved in protein synthesis as it is going to be part of a structure involved in translation. So looking back, when we were talking about carbohydrates, monosaccharides were the monomers of carbs, proteins were made of amino acids, and now looking at nucleic acids, the monomers of DNA and RNA are nucleotides. And nucleotides are made of three different parts. Here's one nucleotide over here. The first part is the nitrogenous base. Then we have our pentose sugar, and we have one or more phosphate groups attached to the sugar. Looking more closely at the nitrogenous base, which is an organic molecule with CHON, remember carbon, hydrogen, oxygen, and nitrogen, we can divide these into two groups. One group is known as the pyrimidines, and these are shown up here. These are single ring bases that include cytosine, thymine, and uracil, which differ in their functional groups. And then the other are two ring bases known as purines. These include adenine and guanine. And one of my former instructors taught me this a long time ago, that to remember these two groups, we could remember pyrimidines kind of sounds like pyramids, like a pyramid shown here. Pyramids are kind of sharp at the edges, or the points, I should say. So you can think pyramids cut, cytosine, uracil, and thymine. Purines they taught us pure silver. The atomic symbol for silver is Ag, so you can remember that as pure silver. And then when I'm looking at the sugars, the purple part here, the type of sugar you have in your nucleotide really depends on if you're looking at DNA or RNA. So DNA, you'll have deoxyribose as your pentose sugar. In RNA, you'll have ribose as your pentose sugar. And just as I asked you to be able to draw a generic amino acid when we were talking about proteins, you should also be able to sketch a generic nucleotide for something like one of our tests. So if I wanted to start drawing one, I almost always start by drawing my pentose sugar, and I'm going to make it generic. I'm not going to label anything. These are supposed to be straight lines. Sorry, shaky hands. So that's my pentose sugar. I know I have a base, a nitrogenous base. So I'm just going to write base. That's my nitrogenous base. And I know that can be A, C, T, or G, or even U if I'm looking at RNA. And then I know that I have a phosphate group attached to the sugar. For the sugar, I know from chemistry that the points at the edges over here, these represent carbon atoms, and this is carbon number 1. This is number 2, 3, 4, and 5. And we actually say 1 prime. That little apostrophe stands for prime. 1 prime, 2 prime, 3 prime, 4 prime, carbon 5 prime, to differentiate it from the carbons that are found in the nitrogenous bases. So there we have a generic structure of the nucleotide. All right, so what are some differences between DNA and RNA besides that ribose sugar? Usually DNA is double-stranded, and we'll see something called a double helix. And usually RNA is single-stranded, although there are exceptions in viruses. There are some viruses that have single-stranded DNA and double-stranded RNA. There are also some exceptions in our own cells where the RNA structure is not double-stranded, but parts of it will fold up on itself and hydrogen bond with itself. All right, we said there were differences in the pentose sugar. DNA has deoxyribose, whereas RNA had ribose. What's the difference in the sugar? If I look at these two, it really lies in carbon number 2. So if I look at what's attached to carbon number 2, in DNA, it's a hydrogen atom, whereas in RNA, it's a hydroxyl group. And this actually makes RNA less stable because hydroxyl groups are more reactive. More reactive, meaning less stable compared to DNA. If I look at the nitrogenous bases, there are also differences here. In DNA, we have adenine, thymine, guanine, and cytosine, whereas in RNA, there is no thymine. Instead, we'll find uracil. Last note here, it's not really a difference, but whenever you're building a polynucleotide using these nucleotide monomers, you're going to be adding nucleotides to the growing chain by adding on to the 3' end of the pentose sugar over here. So if I wanted to add another nucleotide and build, I would add here and then add another one and add another one, always continuing the growth at the 3' end. You cannot add to the 5' carbon because it's already attached to a phosphate group. You're not going to knock that off to add something. So again, we're only going to add to the 3' end. So let's say we have our typical structure of DNA, which is in its double-stranded or double helix structure. Usually what this looks like is a twisted ladder. If I have a ladder here with the rungs over here, if you could twist it, if I could make this an animation, and I twisted it, I would see the double helix. The backbone are the sides of the ladder, and I'll make them dark red here. That's known as the backbone, and I see them here in greenish blue, that zigzag over here, and then also an orangish red color. And the backbone is comprised of the sugar and phosphate components of the nucleotide. The nitrogenous bases, on the other hand, are in the middle. they represent the rungs of the ladder. So this is where you see the A, T, G, and C of DNA. It's also interesting to note that the backbone, when we're looking at the strands of DNA, the double strands, they are anti-parallel. They run in opposite directions. So what this means is one strand will run from 5' to 3', whereas the other strand will face the opposite direction, 5' to 3'. So you can see these are opposite. They're not facing in the same direction, and again, that's called anti-parallel. And the reason that DNA can be double-stranded is because the nitrogenous bases that form the rungs of the ladder here are going to be hydrogen bonding to the base on the opposite side or the opposite strand. And here's another look at that base pairing of these nitrogenous bases. So if I'm looking at a partial segment of double-stranded DNA here, the sides are the backbone, the sides of the ladder that we just saw earlier, and I can see it's made of phosphate and that sugar, phosphate, pentose sugar, phosphate, pentose sugar, etc. And here are my bases, thymine, adenine, guanine, cytosine, and I can see that the base pairing is through the formation of hydrogen bonds. Here our labels are found, 5' to 3', 3' to 5', so I can see that they're running anti-parallel to each other for the two strands. And if I look carefully at what bases pair with each other, A always pairs with T, and G always pairs with C. Or you could say it the other way. You could say T pairs with A, and C pairs with G. The number of hydrogen bonds is also indicated in this picture, and also I try to do that in the parentheses here. There are always 3 hydrogen bonds between G's and C's, and 2 hydrogen bonds between A's and T's. And because of this, if you have DNA and it has a greater percentage of G's and C's compared to A's and T's, that would actually make it a little bit more stable because of the greater number of hydrogen bonds. So if we understand the base pairing rules in DNA, if we have only one strand of DNA, such as the one given here, we can create the second strand. We'll know what the other side is going to be. And earlier I mentioned we always build DNA, build polynucleotides by adding to the 3' end. So I'm actually going to start on the right side. I'm going to start with the 5' end where the phosphate group is, and I'm going to build from there. So I know C always pairs with G, so that's going to be a G here. That's going to be C, C, G. A always pairs with T. This is going to be A, and T, and this will be my 3' end. Additionally, if you understand the base pairing rules, that can give you a heads up as to the composition of DNA if you're given the percentage of one nucleotide type. So if I know a double-stranded DNA molecule is composed of 30% cytosine bases, then I know C always pairs with G, so G must also be 30%. And that adds up to 60%. I know there's 40% left, and I know the percentages of A's and T's has to be the same, so that must be 20%, and that must also be 20%. There are actually 4 types of RNA, and earlier we talked about the first 3, messenger RNA, ribosomal RNA, and transfer RNA, and their involvement in protein synthesis. There's actually one more type called microRNA, which we'll talk about more later on in the course. These are the smallest RNA molecules, and they're going to be involved in the control of gene expression, whether DNA is allowed to be read and eventually produce proteins or not. So how do we get from DNA to protein? The first part is, or I should say the first step, is transcription. You read DNA and you make messenger RNA. Just like the double-stranded DNA that we saw earlier, where if you're given one strand, you can figure out the sequence of the second strand, the same is true when you're synthesizing messenger RNA. Except, remember, in RNA, instead of thymine, we have uracil. So if you read A and you would otherwise pair it with T through base pairing rules, instead of thymine, you would put down a uracil. So what do I mean by this? Let's say I want to go through the process of transcription right here. And I'm given my DNA sequence, one strand of my DNA. How do I figure out what my mRNA molecule is going to be? And sometimes we call it a transcript, which is your product of transcription. So just like before, we can only build from adding nucleotides to the 3' end. So I'm going to start with a 5' end here. And this is my messenger RNA. So if I read cytosine, I know it's going to pair with guanine and vice versa. Here's guanine, G pairs with C, T pairs with A, T pairs with A pairs with T, but wait, I'm looking at RNA. So A, here I'm going to put U, uracil, instead of thymine. And then my 3' end here. So what happens after we make our messenger RNA? The next step involves the process of translation, and we're going to see it's a multi-step process, and I'm going to show you a picture that summarizes the phrases here. So we have our messenger RNA, and the next thing we want to do is start building our polypeptide chain made of amino acid monomers. How do we do that? A large protein structure called a ribosome is going to come into play. And ribosomes actually have two subunits, a larger subunit and a smaller subunit, and both pieces are made of ribosomal RNA plus proteins. So what will happen is our messenger RNA sits between the two subunits of the ribosome. And we're going to have again, this is our messenger RNA. We're going to have tRNA molecules come in, and tRNA molecules are going to recognize 3 bases on the messenger RNA that are clustered together into something known as a codon. So 3 letters on the mRNA, such as CCG or AUG, when they come together, these are called codons, and each of these is equivalent to something called, or it's going to be equivalent to or encode an amino acid, and they each encode usually unique amino acids. The tRNA is able to bind to the 3-letter codon on messenger RNA because it's going to have a complementary set of nucleotides on one end of the tRNA molecule. On the other side of the tRNA molecule, the transfer RNA molecule, you'll see the amino acid that corresponds to the codon. And we're going to see this in a lot more detail in a future lecture. Our book provides us a nice little table to summarize some of the key differences between DNA and RNA. So we know DNA carries our genetic information. It contains the instructions to produce all of the proteins and some of the other molecules like RNA in our body or in cells, whereas RNA is primarily involved in protein synthesis. Where do we find these? DNA is in the nucleus of eukaryotes. It's going to be in the cytoplasm of prokaryotes. And RNA is able to leave the nucleus in eukaryotes. DNA is usually double-stranded in that form of the double helix. RNA is usually single-stranded, although we said there are some exceptions in viruses. The sugar in DNA is deoxyribose. In RNA, it's ribose. The pyrimidines we see are cytosine and thymine in DNA, whereas we have uracil in RNA, and the purines are the same in both. And I just wanted to make a note that even though RNA is usually single-stranded, with exceptions that I mentioned earlier in viruses, there are many RNA molecules that actually will form 3-dimensional structures even in our own cells. And this is formed by hydrogen bonding with itself, folding up upon itself to form some kind of 3-dimensional structure that is critical to their function inside of our cells. Looking at DNA and RNA, and we briefly talked about how these are involved in protein synthesis, this picture shown here, the process of going from DNA to RNA through transcription and going from RNA to protein through translation, is known as the central dogma of molecular biology, or sometimes the central dogma of life. DNA can make copies of itself as well through a process of replication. But this part, how we produce proteins, is really the central dogma, and it was first stated or articulated by Francis Crick. There are some developments in biology, you could say discoveries in biology, over the last 4 decades that has slightly changed our view of this. So we used to think that it's not possible to go backwards, right? But now we know that there are some viruses, such as human immunodeficiency virus, HIV, the virus that causes AIDS, that are in a group called retroviruses. Interestingly, these retroviruses contain an enzyme called reverse transcriptase. I'll abbreviate it RT for short. It does what it sounds like, the reverse of transcription. So these viruses, they have this enzyme, and they can take RNA and go backwards, turn RNA into DNA. And that's actually what HIV does. If it infects human cells, it injects its RNA, uses its own reverse transcriptase to read RNA and convert it into DNA, and that DNA will insert itself into our own DNA and hide itself there. We can also use reverse transcriptase in the lab on purpose when we want to clone or copy DNA. And we'll see how to do this at the end of the semester as well. All right, that takes us to the end of the 5th and last video for Chapter 3. Next time, we'll start Chapter 4, which looks at the cell, the smallest unit of life. And this will include both prokaryotic and eukaryotic cells. All right, thanks so much for listening, and I'll see you next time.